In the second part of the presentation, we will discuss about non-regulatory in vitro assays used for dermal safety and efficacy testing. I would like to address 3 assays that can be used for non-regulatory in vitro safety assessment: the time to toxicity screening assay, the single short exposure and cytokine analysis assay, and the hybrid assay, which is a mix of the skin corrosion assay that you have seen before and the time to toxicity. Finally, I would like to discuss efficacy assays that can be performed in vitro for anti-inflammatory and skin lightening efficacy results. I will start with the non-regulatory in vitro safety assays for dermal irritation and corrosion. They are usually performed to support product development, research and development, and innovation groups within industry for the evaluation of prototypes, safety, and efficacy. The assays address multiple endpoints: primary or secondary cytokines, histology, and other endpoints of interest that provide additional insights into the safety profile of actives or finished formulations. These type of assays can also be used using multiple exposure times that offer the possibility to compare the irritation potential of candidate formulations for the ranking of irritation potential. This is especially useful when using benchmarks or when reformulating products towards a less irritating profile. If we think back of the regulatory assays that I presented in the first part of this lecture, they were using a single endpoint and a single possibly cutoff value for the endpoint and possibly 1 or maximum 2 exposure times. With those limitations in mind, the outcome of the assay would address a yes or no type of question for the irritation of products. With non-regulatory in vitro assays, the horizon of interpretation of the data is widened. If we look at the framework of industry, it is important to know where each player plays a role. So industry comes into the picture with various concepts on ingredients, natural products that have to be tested for safety or for efficacy before having a finished product put on the market. So it is a long way from the concept of the product to the product launching, and even after that into post-market follow-up. So for the raw ingredients or the finished formulation, efficacy testing and safety testing should be performed. And the assays that can be used address various endpoints: skin irritation, skin sensitization, or anti-inflammatory action, or skin tone modulation. It is a back and forth between the efficacy testing methods and safety testing to provide a preclinical profile of the ingredient of interest or a final formulation. There are multiple types of assays or test systems that can be used to address the preclinical in vitro testing, either using co-cultures, cell-based models, ex vivo models, or reconstructed tissue models. Before every product of interest, particularly formulations for cosmetic industry, even go into clinical testing. So testing labs have heavy work in this area, performing in vitro assays in the setup for the preclinical evaluation of raw ingredients and formulations. There are two types of assays that I would like to discuss together and to emphasize the differences or the aspects that are common between the two. The time to toxicity assay, or the screening assay, uses reconstructed tissue models for skin and the same endpoint of tissue viability addressed by MTT. We calculate the % viability of the tissues, but also what we call an ET50 value, or effective time, which is the time that it takes for a product to reduce the viability of the tissues they are exposed to by 50%. Occasionally, using this type of assay, histology may be performed on the tissues. Like with any assay, we use specific negative and positive control. In this case, Triton is the positive control, which is a surfactant. The assay, the time to toxicity, allows toxicity profile comparison between test substances of interest and also ranking of irritation potentials. The assay is also used for qualifications of benchmark materials. I cannot emphasize enough the criticality of using benchmark or reference materials in studies, particularly with this type of non-validated assays. In other words, it is important to include with a prototype a benchmark material that is fairly similar in chemistry or in impact onto the tissues in the study, because the data on the prototype should be compared to the data generated from the benchmark material rather to be compared to the assay negative or positive control, which may not be in the same chemistry range. The other assay that is also used with the reconstructed tissue models and the same endpoint of MTT tissue viability is what we call the single short exposure plus cytokine analysis assay. This was generated particularly for rinse-off products, shampoos, conditioners, and similar chemistries that were relatively mild. So the MTT endpoint was not necessarily capable to rank order the irritation potentials of this product. Because of that, a second endpoint was introduced in the assay represented by interleukin-1 alpha. And it is very important to look at both sets of data of tissue viability per MTT and cytokine expression by interleukin-1 alpha when analyzing the profile of a product subjected to this test. One of the limitations for these two assays is that a correlation between in vitro and in vivo or clinical data needs to be generated through repeated testing to address appropriate cutoff values for specific chemistries of line of products. In other words, it is the duty of the manufacturers to build these programs internally and to have their own program to address formulations in particular if the validated assays are not necessarily needed for regulatory purposes. The time to toxicity or screening protocol is performed as follows. The tissues are incubated in fresh media for about an hour. The materials are applied on the top of the tissues, mimicking a topical exposure, anywhere from 1 to 24 hours. The volume that is used for these materials is bigger than the volumes that was used for regulatory assays. It is 100 microliters. At the end of the exposure, the tissues are rinsed and the MTT reduction starts. It's followed by the extraction, isopropanol plating onto a 96-well plate, and we generate optical density values as with previous assays. The differences between the screening protocol, which can address leave-on products or rinse-off, and the single short exposure and cytokine analysis assay are as follows. In general, since cytokines are analyzed in this assay, the tissues are incubated overnight for approximately 16 hours in hydrocortisone-free media. It is the purpose of this assay to not impact the tissues and to allow them to express the cytokines. So hydrocortisone is removed from the media for the purpose to allow the tissues to build up the cytokine expression. The tissues continue that incubation in fresh media for about an hour before being treated. The tissues are treated with the test substances, are rinsed after the exposure, which is normally 1 hour, and they are added into new media for a post-exposure time of 24 hours. This post-exposure time is necessary for the expression of cytokines. The media is collected after the 24-hour post-exposure, and it is analyzed using an ELISA assay specific for interleukin-1 alpha. How are the data interpreted and analyzed for the time to toxicity assay? Usually, the data are presented in what we call response curves, where the tissue viability is plotted against the exposure time that was used for a particular product. For example, for this product in green, where the 50% viability is not even achieved after 24 hours of first exposure, we can say that the profile is probably of a non-irritant product to the skin. This product reached its 50% induced a 50% reduction of tissue viability around 10 or 11 hours. So it could be a moderate irritant to skin. Whereas the product in red, it's probably a corrosive or a severe irritant because the ET50 value was achieved probably in the first couple of minutes of exposure. As I said before, there is no prediction model generated for this type of assay. So the correlation of results with expected in vivo results has not been firmly established. However, for example, one of the manufacturers of tissue models, Meta Corporation, suggested this guideline, if you will, for assigning verbal descriptors for expected in vivo irritation based on the ET50 values obtained with their tissue model. As you can see, for example, the assay positive control, Triton X100, could be considered a moderate to mild ingredient if the ET50 values fall anywhere between 4 and 12 hours. Whereas nitric acid is a potentially corrosive with an ET50 value of less than 0.5 hours, similar possibly to this product, hypothetical product here. Other products, such as the baby shampoo, was very mild because the ET50 value was anywhere from 12 to 24 hours. I would like to give an example of pure chemicals that were tested using the time to toxicity assay. This is a critical work that has been performed a couple of years back as part of a pre-validation work when industry was trying to design the skin irritation test, which I presented in the first part of my lecture. So the time to toxicity assay was considered at the time with the goal to address all GHS categories. The task was more challenging than one would have expected, and the tissue models that were used with the chemicals included in the pre-validations had a high false negative rate and some other caveats. As you can see in this table, some of the products, whereas in vivo they were not predicted to be irritants, they were predicted irritants in vitro. So it was concluded at the time that the time to toxicity assay may not be working for regulatory purposes, and that's how industry moved towards a single exposure, a single cutoff value for the classic validated skin irritation test. Nonetheless, the time to toxicity assay is heavily used in industry, particularly for products that may be prototypes, are not regulatory submitted, again, to address product development or innovation purposes within companies. One example of the use of the time to toxicity assay is covered in the work by SCJ. I referenced this poster in the first part of my presentation for the comparison of corrosive assay. In this exercise, mixtures across all GHS skin categories, skin irritation categories, were used in a time to toxicity approach using exposure times anywhere from 3 minutes to 480 minutes, with the goal to address all 4 GHS categories. So what was concluded after this exercise was that the epiderm time to toxicity assay did not distinguish among all GHS skin categories, but provided a clear cutoff value of 15 minutes or less for the GHS category 1 mixtures. This is very important work because it addresses mixtures, not ingredients, which were classically used for to validate in vitro assays. As you can see on the top part of this graph, the other categories, category 2, category 1, and category 2, 3, and no categories were all clumped together on the top of this cutoff value of 15 minutes. Another example comes from cosmetic industry using volatile organic compounds, or VOC, containing products. This is work by Alberto Culver at the time that was presented in 2009. The company has shared data, clinical results for skin, RIPT, and they shared that there were not so many consumer complaints on this products included in the study. However, when the classic 100 microliter dose was used for the epiderm-based time to toxicity assay, we calculated very short ET50 values, which would be indicative of irritation potential. However, clinical data and consumer experience data did not state like that. So through discussions with the sponsor at the time, it was identified that possibly the 100 microliter dose would not be the appropriate one. And we repeated the studies using a much smaller dose of 30 microliters, which is, again, the dose that was used in the skin irritation validated assay, just to have a point of reference. In this situation, as you can see, the ET50 values obtained for the product for more than 24 hours, and they were much aligned with the clinical results. So it is, again, very important to know the chemistries if possible, and to modify assays if there is such flexibility in such a way that they better predict a clinical outcome. And in this way, products can be actually ranked from high irritation potential to low irritation potential, and be compared to references, if you will. In this case, ethanol, a different concentration was used as a point of reference because there was also an ingredient in the products tested, so relevant to the chemistry. Moving into the second assay that I would like to present, the single short exposure and cytokine analysis, I would like to emphasize that the assay has been designed for surfactant-containing products that were very mild, in the range of baby shampoo type of products. And this is work that has been performed in collaboration with Johnson & Johnson. Again, this is public domain information. We published a manuscript together a couple of years back in 2016. And in that work, we tested products that, in terms of viability of the tissues used, the epiderm tissue model, they were mostly non-irritant or mild. So they were all kind of grouped in this area of the tissue viability. So we couldn't really compare any irritation potential that may have occurred. So the second endpoint that was used for the assay was the interleukin-1 alpha, and you can see a better spread of the irritation potential that the company could use to rank the product. The work presented in these graphs was based on formulations. However, the company was also interested to address individual surfactants and there are different concentrations that can be used in formulations to see which classes are more irritating than others. For example, we know that this type of surfactants are very irritating because the value of the IL-1 alpha that is secreted from the tissues treated with them is very high. Finally, the interleukin-1 alpha data that were generated in vitro were correlated with the transepidermal water loss, which is a clinical endpoint. And the correlation was deemed to be satisfactory for the purpose of the company in addressing skin irritation events. The next assay and the last of the non-regulatory safety assays that I would like to briefly present upon is called a hybrid assay, which uses reconstructed tissue models and the same tissue viability endpoint using appropriate negative and positive control. The assay is a mix of the corrosion assay that is validated for regulatory purposes and that you heard about already, and the time to toxicity assay that I just presented about. In terms of this assay, usually for the applicability, it allows the separation of corrosive and non-corrosive materials since it has a corrosion component. But from the time to toxicity standpoint, allows toxicity profile comparison between test substances of interest and ranking in the same assay. It is important to have the same tissue lot to address multiple endpoints, possibly in the same time concurrently. One of the limitations are the same with the corrosion assay regarding the separation of subcategories 1B and 1C. Whereas for the time to toxicity, we already know that the correlation with the in vitro, between in vitro and in vivo and clinical data should be performed by the companies before using this platform as a reliable one. Here is how the assay is performed. Usually, the tissues are exposed to the test materials for 1, 4, and 8 hours using the time to toxicity assay and dose of the material of 100 microliters. If the tissues are observed when transferred into the MTT endpoint to show toxic effects at 1 hour and 4 hours, it is determined that there is a potential for corrosion activity. So the corrosion assay is going to be performed as per the OECD guideline for 31, and the material can be addressed as corrosive or not. If there are no or minimal cytotoxic effects observed at the tissues treated for 1 and 4 hours with the materials, then the time to toxicity assay is continued with another exposure time of 16 hours. And between these 4 time points, an ET50 value can be calculated and interpretation of the data to the irritation potential can be made. The advantage of this assay is that it can address irritation with a screening component and corrosion in the same time using the same lot of tissues. So let's review what we learned thus far. Coming back to the DRAIS range of irritation, remember that we talked about what assays addressed what type of irritation potential. So we addressed corrosive materials and irritants. In the middle range, for which we had a question mark before, we believe we can address moderate irritants using the hybrid assay. And then in this range, we moderate to mild to non-irritant, we can use the time to toxicity assay. And then at the end of the spectrum for very, very mild or non-irritant products, we can use the single short exposure and cytokine analysis assay. So in this way, using multiple in vitro assays in different fashions, we can address the entire range of irritation potentials as per DRAIS animal tests. And this is how we can put this in perspective, or in a lab perspective, if you will, in terms of MTT response, where non-irritant materials would not affect the tissues, which are capable to reduce the MTT, whereas a corrosive material would be so damaging to the tissue that it's not capable to reduce the MTT. In this part of the presentation, I would like to discuss efficacy assays that are used for anti-inflammatory action and for skin tone modulation activity. Let's start with anti-inflammatory assay. The test systems that can be used is, again, reconstructed tissue models. Any skin model can be used. The inflammatory state can be induced by a number of agents. However, we have experience working with PMA, and we need to be aware that cytotoxicity of these inducing agents should be low, not to confound the data. The assay endpoints are tissue viability using MTT endpoint and cytokine analysis using various cytokines, either IL-6, interleukin-8, TNF-alpha, etc. It is very important for the products going into this assay that they are not toxic. Since cytokine analysis is the primary endpoint, it is important that the products are mild or non-irritant, not to force the tissues to produce cytokines aside from the irritation that we induce to them. The assay negative control in this case is saline. It's physiologically relevant to the process of the assay. And the positive control could be a hydrocortisone type cream or equivalent. Here's how the assay is viewed in terms of data result and interpretation. The products are applied on the top of the tissue, and the inflammatory agent is added to the media. When it is a competition, if you will, between the PMA, the inflammatory inducing agent, is trying to exacerbate the production of the cytokines, whereas the product that is designed to reduce the inflammatory events does the opposite. The exposure time is the same, it's 6 hours, and this is how the assay is performed. We use an overnight incubation in hydrocortisone-free media just to acclimate the tissues and to allow them to stay in culture a little longer. We use a set of 3 tissues cultured in media containing PMA and another set of 3 tissues in media containing ethanol as a control. Ethanol is used to dissolve the PMA. The same steps are used afterwards, where the tissues are rinsed, added to MTT, extracted, and then an optical density value is calculated for the viability. The media is collected separately and used in the cytokine analysis. I would like to give an example of how the data are interpreted. This is hypothetical set of data using interleukin-8, where the red bars represent the IL-8 production of tissues cultured in media containing PMA, and in the blue in media not containing PMA. So what we're looking for is to see that in the negative control tissues, the PMA induces a spike in the production of the cytokines. That means the test system is responding to the inflammatory agent. What we also look for is that the positive control, which is a hydrocortisone cream or any anti-inflammatory cream, manages to reduce this production of IL-8 or any cytokine of interest to a low level. That means the positive control is efficacious. If we're looking at these 2 hypothetical test materials, we can see that this material is actually very efficacious in keeping the IL-8 production relatively low, whereas this one is not as efficient in reducing the inflammatory events. I should state that the cytokines or chemokines of interest depend on the pathways that the prototype of the anti-inflammatory product is targeting. So it should a discussion between manufacturer and the testing lab should take place in determining the best cytokines to be used, usually secondary cytokines, and we find that IL-8 is probably the better expressed in the tissues. The examples that you will see in the next slide are generated using the Epiderm model from PETA Corporation manufacturer. In this study, we used over-the-counter and prescription pharmaceutical products with anti-inflammatory action, and we tested the media collected from the tissues for the interleukin-8 endpoint and for the interleukin-1 alpha. There are 2 differences between these 2 sets of data. The IL-1 alpha was generated using the lysed tissues, presumably because there is an overload of interleukin alpha in the tissues. and IL-8 was analyzed from the media. The key information here is that it takes 6 hours or more for the interleukin-A to be secreted in the media, whereas interleukin-1 alpha needs 24 hours of post-exposure to be expressed and released into the media, if you remember from the cytokine assay that I explained before. So what we can see in this set of data is that this is the saline, the negative control, and we can see a large amount of IL-8 that is produced in the tissues. The over-the-counter anti-inflammatory cream reduced this level by about a half, and the prescription drug reduced it even more. So the test system is fairly sensitive in ranking, if you will, the potency of anti-inflammatory products, where intuitively we would think that a prescription drug is more potent than an over-the-counter. The same can be stated for interleukin-1 alpha. However, the values of the cytokine are much lower compared to the interleukin-A, which is considered a good marker for this assay. The next efficacy assay that I would like to discuss is an optimized melanin modulated screening assay that it can be used for skin lightening effects or darkening, depending on the goal. The test system is again based on reconstructed human epidermis models that contain melanocytes from different phototypes, either Asian, African American, or Caucasian. These are the providers that are around the world: Cell Systems in Germany, Episkin in France, Mate Corporation in the US, and Tigo Science in Korea. I will concentrate my presentation on this model called Melanoderm from Mate Corporation, and specifically the Asian phototype, because we generated data and we have more experience with the system. The assay endpoints are tissue viability, again addressed by the MTT endpoint, the melanin concentration, histology that uses either the regular hematoxin and eosine for the architecture of the tissues, or Fontana-Mason, which is specific for melanin production. Macroscopic and microscopic pictures are also key for this type of assay. In general, we use specific negative and positive control. The positive control is 1% kojic acid, which is not toxic at this concentration, and for the assay we optimize, and is known to be an inhibitor of the melanin production, specifically of the tyrosinase, which is the key enzyme of the pathway. The assay is used in support of product development, new technology, and innovation projects for screening of active finished formulations with potential to modulate melanin production and impact skin tone. One of the limitations of the assay is that there are small differences in melanin production between tissues treated with various actives, depending on the phototype used. Courtesy to Mate Corporation, I would like to emphasize how the melanocytes look like in an African American phototype tissue model, and how the tissues look like being darkened in culture up to day 21. The assay can be performed for 1, 2, or 3 weeks, and it has 2 parts. One is a dose range finding that uses 2 different volumes of the product, 10 and 25 microliters, and the other one is a definitive assay that uses the melanin endpoint in addition to the viability. The dose range finding is very important to ensure that the products are not toxic to the tissues, such that any reduction in the melanin production is not due to toxicity. Melanin reduction should be as a result of the product impacting the melanogenic pathway, but not of the cytotoxicity per se. This is how we optimize the assay. We performed it for 1 week, 2 weeks, or 3 weeks, and analyzed the production of the melanin in negative control treated tissues and positive control treated tissues. We noticed a difference after 1 week with a ratio between the negative and positive control of 1.5, which was even higher after 2 weeks, and about the same after 3 weeks. So we thought of many reasons that the assay can be performed for 1 week to optimize the assay, to avoid any significant differentiation of the tissues in culture for up to 3 weeks, which may impact the penetration potential of the product as the time in culture goes longer. So the optimized assay that we perform uses 4 treatments every other day. And at the end of the treatment, the endpoints, viability and melanin endpoints are performed. I would like to show you macroscopic pictures of the tissues. These are Asian melanoderm tissues untreated at day 0, where you can see that not much pigmentation is visible. And these are the negative control treated tissues and positive control treated tissues after 7 days. You can notice that the positive control treated tissues are possibly at the same level of production of melanin like the untreated tissues at day 0, indicating an efficacious positive control, whereas negative control treated tissues are much darker in comparison. These are African American phototype pictures, much darker at day 0 compared to the Asian. Definitely much darker after 7 days. This is the negative control treated tissue, and this is the positive control treated tissue. Macroscopic pictures are also important to look into the health of the tissue and any morphological changes in the cells after treatment with the products of interest. These are untreated tissues at day 0 for the Asian phototype and African American phototype, where you can see that the melanocytes show visible dendrites that are much darker compared to the melanocytes in the Asian phototype. As the time in culture goes by, the tissues darken in the negative control treated tissues for the 2 models. This is the Asian and this is the African American, more darker in comparison. This is the positive control tissues showing actions of the control onto the melanocytes production of melanin. Finally, when African American phototype tissues are kept in culture for an additional week, you can see how dark they become in culture. One of the endpoints that are used in the assay is tissue viability. So if we look at the tissue viability values versus time in culture, and considering the negative control treated tissues 100% viability by default, we can see that the positive control, kojic acid, is not toxic after 7 days in culture at 1% concentration. As we increase the concentration of the positive control up to 5%, we can see that it becomes cytotoxic even after 7 days. If the assay is performed for 14 days, for example, with the positive control, even 1% tissue viability shows toxicity effects versus the 1-week assay, and the 2% is even more toxic. We'll also notice with longer exposure times variability between the tissues, possibly due to differentiation of the tissues and thickness of the stratum corneum, that may impact the rate of penetration of the positive control through the tissue. So this assay, if it's used in a pre-screening manner, can help in deciding whether the product should go into a definitive assay or what volume should be used. For example, if a 70% viability of the tissues is observed for both 10 and 25 microliter dose, then the 25 microliter dose should be used in the screening assay, and so on and so forth. If the viability is really low for the 10 microliter dose, the material is likely an irritant, and its action on the melanocytes from the cytotoxicity standpoint should be further analyzed. The other endpoint of the assay is the melanin endpoint. And comparing the production of melanin again against time, we can see that the production of melanin in untreated tissues in microgram per milliliter is relatively small. The negative control after 7 days has a much higher production of melanin, and the positive control induced a reduction in melanin almost close to the untreated tissues. If the assay is continued for another week, up to 14 days, we can see a significant increase in melanin production in negative control treated tissues. However, this is not the case for the positive control at 1%, which shows that toxicity is involved in the reduction of melanin at this point, confounding the efficacy interpretation of the data. There are a couple of factors to be considered when transitioning from cell-based to tissue-based assays and in preparation for clinical studies. There is a different penetration rate of actives in these test systems. They may be more readily available to cells rather than to tissues, rather than to human skin. So bioavailability should be taken into account. Tissues differentiate in culture, as I mentioned already 2 or 3 times, and toxicity versus efficacy of actives that impact melanin production is key to data interpretation. So, coming back to what industry is facing when putting a product on the market, it is a lengthy and complicated process to translate a concept into a product. Coming from an idea of a prototype and going into safety and efficacy testing, multiple possibilities, in vitro possibilities to address these goals are available on the market. Any of these tissues can be used for efficacy and safety testing to ensure that the consumer is not exposed to adverse effects when using a finished product. And we all, whether we come from industry, regulatory, or testing labs perspective, we all contribute to this goal. If we consider again our work together in sharing the same perspective, challenges, and common goals, I think we can state that we all work for the same goal of ensuring that the consumer is safe. And from the perspective of cosmetics products and other products that are regulated by FDA, it is to be commended the effort of the agency, particularly this year, that put out there the FDA's Predictive Toxicology roadmap. That roadmap takes into account a couple of parts where all these players will come together, will be trained or will train in addressing other methods that can be used in place of the animal test, either in silico, in vitro, ex vivo, etc. And from that perspective, it is also commended to see, and it is a big step forward to see that Taiwan banned the cosmetic animal testing, as stated in this document 2 years ago. So all parties are coming together in working to replace animal testing, and there is much more work to be done, and to advance the field and to look for relevant methods that in the end can replace the use of animals. I would like to thank all the parties who contributed to this presentation today. Thank you for the invitation from the PETA International Science Consortium, and also to all of you being part of this lecture, and to my team at home, the Institute of In Vitro Sciences. Thank you very much for your attention.